So now let's look at the third problem for hidden Markov model. It's a harder problem, that's why I grade this out. We are not going to ask you to really learn all the details, but I'm hoping that you will get some intuition behind it. Okay, so this third problem is we are given the observed string of coin flips. The coins that are used is still hidden, but also all the parameters in this hidden Markov model are also not given. And we have to estimate the parameters and also make guesses of what coins are being used. So you can imagine this is a much harder problem. And the solution for this is called the Baum-Welch algorithm. It's similar to the expectation maximization algorithm, which are used for transcription factor motif analysis. So the idea is we randomly initialize some parameters for the hidden Markov model. And then based on the observations and this randomly assigned parameter, we try to guess the hidden states underneath all the coin flips. And then based on the predicted states, the hidden states, the coins we use, then we can update the parameters in the hidden Markov model. And then we iterate this process, hopefully that it will converge onto something. But imagine if you have a solution landscape like this, depending on where you start your random initialization, if you start from here, it will give you a local maximum. If you start from here, it will find the local maximum. If you start from here, it will get there. If you start from there, it will give you a local maximum, but there's no guarantee that you will get the global optimum solution. And so this one you need to restart a number of times. So let me show you roughly how this is done. So supposedly we are in the coin flip example again. You are given a string of coin flip. In order now to estimate the parameters, we need to make sure that the string of observations are very, very long. If you don't have enough observations, you won't get a good parameter estimate, right? So initially, we randomly start something. So what we do is we do just like use your eye, make some intelligent guesses. What are the coins that we assigned? You can see here, okay, these are, roughly 50-50. So we can randomly, not really random, but like we can, assign them probably a fair coin. But you probably notice, okay, around here, these are mostly heads, so it's likely to come from a biased coin. So you assign them kind of biased. And these ones, you're not so sure. So, maybe fair, maybe biased. But so imagine you have a very, very long string of coin flip. You make some reasonably intelligent guesses. And that's your initialization. But once you assign a coin underneath every of this flip, we can now estimate the initial probability. How do we do that? So the probability of using a fair coin is we ask, out of all the coins we assigned, what is the total number of fair coins we have over all the coins that we have, right? That's your initial probability. What is our transition probability? You ask, okay, so we have fair coin, biased coin. You ask, from one coin to the next, you see how they transition. How often do we see from a fair coin, the next one, I'm still using a fair coin? How often do I go from a fair coin now changing to a biased coin, right? And how often I'm in the biased coin, the next coin is still biased, and how often I transition back to the fair coin. And you can just use the current initial assignments to estimate those parameters. And how do I assign the emission probability is you ask, if I'm using a fair coin, what is the probability? Or you can see whenever I'm on the fair coin, what percentage of those fair coins are under a head, and what percentage of them are under a bias coin. Under a tail, giving out the tail. And then the emission probability for the bias coin is similar, you ask under those or on those biased coins, how often do I see a head and tail? And so that gives you the initial parameter for this hidden Markov model, right? And so now with the parameter, let's forget about the initial assignment that you gave each coin. We just, we take it out. We use the current probability, the current parameters, initial probability, transition probability, and the emission probability to now use the second problem, right, to make guesses of what's the coins that are used under each of the observations. You can say use a forward-backward algorithm. You calculate all the coins that's used, and after you have now a new path of, based on the parameters, you estimated, this one should be actually a fair, that's a bias, and so on. And based on that new assignment, we can now update, okay, what is now the total number of coins assigned as fair coin? And then based on this new path, what is the transition probability, and how many of those fair coins are giving a head versus a tail? And then you forget about the path again, use the updated parameter to then assign all the coins underneath each of the observations. And so you can see you iteratively update this process, right? And so that's how the solution for the third problem. You can see we can run the forward, backward to assign the path, or the states of each coin, and use those assigned coin to update all the parameters in your hidden Markov model. And eventually you'll get somewhere, but make sure you start a number of times so that hopefully you'll get the optimum solution. Okay. So we mentioned there are a number of hidden Markov models that have been used in bioinformatics. One is for gene prediction. So let's think about what are how do we set the model for gene prediction? By the way, before you come to this lecture, when people say, "Oh, I'm using hidden Markov model," you're like, "Oh, shoot, this is totally out of my league." But now, if you hear somebody say this, you should ask them, "Oh, that's interesting. How do you set your transition probability and emission probability?" And if you ask this question, this person who tell you they're using hidden Markov model will say, "Wow, this guy knows," right? So you show them that actually you know something about hidden Markov model. So let's think about the gene prediction problem. What are the hidden states? What are the time? What are the the initial probability, the transition probability, and the emission probability? So the hidden states in gene prediction is usually for a gene, there's usually a first exon, then there's some other like intermingled introns, exons, and introns, exons, and then from there, then it goes to some intergenic region. And then hopefully then you will start another gene which has exons, introns, and so on, right? So you can imagine a very simplified sequence would be in front of this gene there is some promoter sequence, and there's a first exon, there's a last exon, and there's some internal exons, and there are some intron sequences, and then the rest are more like intergenic sequences. So each of these sequences might have some different properties, right? The different time is nucleotide base, every base, or you can use every 6-mer or every 3-mer to look at this sequence, next sequence, what is the chance that you are in based on whatever sequence you see before and after that you are on an exon versus an intron or intergenic sequence. the initial probability is, oh, you look at some genome, you say what is probability that, say, human genome has 2% coding exon. That's kind of your initial probability. Your transition is, from one base, if I'm on an intron, what is the chance the next base is an intron versus exon or intergenic sequence, right? The emission probability is, if I'm, so if you think about 6-mers as your unit, K 6-mer sequence. If you are in an exon, because they code for amino acid, they will have some property of giving out a certain 6-mers. But if you are in these non-coding regions, they probably don't have those properties because they don't give out protein amino acid sequence, right? So they will have a different emission probability. And so then based on, like if I'm looking at this particular 6-mer, looking at all the sequence before and all the sequence after, I'm trying to guess, is this location in the middle of an exon? Is it in a promoter? Is it in an intron or intergenic sequence? So that's kind of gene prediction. And we can use it for looking at the sequence motifs. And so basically, sometimes you have a whole bunch of sequences, and you are trying to see, they actually, some of them share a pattern together, and they create a sequence motif. And so we are trying to ask if this sequence that I'm looking at is part of the motif or not part of the motif, right? So the state is probably in the motif or not in the motif. The time is, also a string of sequences. The pi is, how often do we see this motif? The transition is from the motif to in the next base. What is the chance you're still in the motif? And also the emission probability is, what is the nucleotide sequence you see in the motif or outside the motif. For protein structure prediction, so this is protein secondary structure prediction. So supposedly we have a string of amino acid sequence, and we're trying to guess, okay, for this protein, maybe this protein have some alpha helix and then a loop and another alpha helix and then some strand, the beta strand, and then some other tail. Okay, so the question is, if I give you the string of amino acid sequence, can you help predict whether this region should be in that alpha helix and that sequence should be in that beta strand and that sequence should be in the loop? Right, so in here, what are the hidden states? The hidden states is alpha helix, beta strand, loop structure, so the different secondary structure. The state, the time is every amino acid to the next amino acid on this string of sequence. The initial probability is you just look at all the proteins we solve the structure to ask, how many amino acid locations are in the alpha helix? How many how often do we see a strand, and how many we see kind of a flexible loop structure? The transition probability is, if I'm already in the alpha helix, what is the chance the next amino acid is still in the alpha helix? What's the chance that it's gonna end, right? And then the emission probability is, if you look at those sequences or the structures that already have structure solved, how many of them do we see different, in alpha helix, what's the likelihood of seeing a proline, a serine, or leucine, and so on? What is the chance of seeing those different amino acid in a beta strand, beta sheet? And what is the probability of seeing those amino acid in the loop, flexible sequences? And so that's for protein structure prediction. We can also use it for chip p-calling, but so you can imagine if I have a region, if I have signals like this, and so maybe there is some noise, right? So you can see, so you might have a little noise in here. so we were trying to see if this is the baseline signal or a increased signal, even though there's some noise. Maybe I don't make a good enough judgment here, but so we're trying to estimate, for example, for this copy number variation, we know in the genome, there are cases when we have 2 copies of the genome, but it could be amplified, and sometimes you might have a gene deletion region, right? So there's 1 copy of the chromosome, so there's 3, and maybe you will even have 4 copies of the genome, or even higher, right? And so the states would be, 2 copy, 3 copy, 1 copy, the different copy number. The time, or is all the signals that you observed across, like a read coverage or something. You try to estimate the copy number. So this is the actual observed sequence that you see on each location. And then we are asking, what is the chance that, say, some cancer genome have this amplification or deletion, and then try to ask, if you have 1 location that's 2, what is the chance that the next base is amplified or deleted? And then the emission probability is, if you are in a 2 copy versus 3 copy or 1 copy of the genome, what's the chance that you see the read covered at these steps, right? So this is another possibility. And so there are many, many ways that we can use this hidden Markov model to understand bioinformatics. So chromatin domains, we will talk about this later in the epigenetics lecture. So in summary, today we talked about hidden Markov model. We first start explaining what Markov chain is. So if you are at the current state, the probability to come here only depends on your previous 1 state, but not the past before that. Hidden Markov model is if, say, in a coin flip, you only observe the final head and tail. You don't know whether I'm using a fair coin or a biased coin. And we try to solve 3 problems here. The first is, if we have the parameters and we observe the string of coin flips, we try to ask what is the likelihood that my model, that whatever the setup, the parameter will actually give you the string of coin flips. And there are forward and backward procedures, and they should give you the same result. The second problem is if we have the hidden Markov model parameter, and we also have the observed coin flips or observed observations, we try to guess the hidden states, right? And so there are 2 algorithms. The backward-forward algorithm will try to maximize the number of correct predictions at each location, whereas the Viterbi algorithm will give you that single path, which is the most likely to give you the whole observation. Of course, a lot of the 2 will give you similar estimates, but they may or may not be identical. And the third is, question is, if I don't even give you the parameters for the hidden Markov model, can you estimate the parameters as well as estimate the hidden states? And there, we don't have an optimal solution. You have to restart a number of times and approximate, get to a local maximum, and that's using the Baum-Welsh algorithm. Okay, that's it for today. I hope you will enjoy hidden Markov model and use it in your machine learning problems.